Okay, everybody, welcome to the last video for this week. Unless I decide to do a exam workshop one, which I probably will. But yeah, just a reminder, your exam is on Friday. At the time of recording today is Sunday, so it's 8 and 5 days. So hope you've been studying, or you're planning on studying, or you feel comfortable with the material. Reminder that you guys get a 4x6 flashcard. And yeah, there's a couple other resources for you on that page as well if you're interested. But today, gonna talk about RTKs, receptor tyrosine kinases, and hedgehog signaling. So let's go ahead and do that. So generally speaking, we've been focusing on paracrine signaling. So we talked about, and I guess synaptic to an extent, acetylcholine stuff, messed around with that. But this is kind of an offshoot of that, where we still are going to focus on paracrine stuff, I would say, but it's a slightly more nuanced topic. So we already talked about ion channels. We spent a couple lectures now on GPCRs. Now we're going to talk about enzyme-coupled receptors, which are, generally speaking, going to be things like receptor tyrosine kinases, although there are other types of enzymes as well. So transmembrane kinases, like RTKs, are enzyme coupled. So what does that mean? It just means that they generally have an enzyme attached to them. So here's an example of a generic RTK. You're going to start with these inactive monomers of the tyrosine kinase. Then you're going to bind your signal molecule, which causes them to dimerize. And once they dimerize, these tyrosine kinase domains are brought close enough together to each other that they actually phosphorylate each other. And once that phosphorylation takes place, it activates more kinase domains, these extra kinase domains, and then we get this thing where we get a bunch more phosphorylations in different spots of the RTK, and these phosphorylations actually serve as anchor points for other proteins to bind and signal to extra or other proteins or molecules in the cytosol. So these things bind and they activate signaling protein pathways. And so what they're trying to get across here is that each phosphorylated tyrosine is neighbored by different amino acids. That should make sense. And those different amino acids kind of decide what proteins are able to bind in these different spots. So for example, this tyrosine, maybe it's surrounded by a bunch of arginines. So maybe the protein that it binds has to have a bunch of positive charges on it, along with a place for the phosphate to interact with. So stuff like that, right? And talked about this, different receptors bind different combinations of factors. So it makes sense, right? This, this RRTRR section is going to bind a different place, or a different protein than maybe this section over here, which is like a TDD or something, which is like glutamic acid and aspartate stuff. So instead, they bind different places. So here's an example of that. So you can see here, this tyrosine is surrounded by a threonine, a phenylalanine, and an alanine. This is going to be binding a different protein than the lysine-lysine-leucine or the isoleucine aspartate proline, right? So these different environments result in different proteins binding to different parts of the RTK. So now we talk a little bit about SH2 domains on the target proteins of RTKs allows specific responses to phosphorylations. So I wish they would show me where the SH2 domain exactly is. But alas, they have not showed it to me. Where is it? That's fine, I guess. Essentially, the SH2 domain is as shown here, if you will. And so I don't know why it says yrosine there, but so there's a binding spot for the phosphotyrosine, which is this spot, and there's also going to be a binding site for specific amino acid side chains. So remember the arginine tyrosine thing. This is the spot where those two arginines would bind, and then the phosphotyrosine would bind here. So the phosphotyrosine really isn't specific for every protein, but the amino acid binding spot definitely is specific for each protein. And so this is specifically the part of the protein that binds to the RTK itself. So, like we said here, trans-autophosphorylation generates binding sites for signaling proteins. It's called trans-autophosphorylation because both of these basically phosphorylate themselves. That's why it's called trans-autophosphorylation. So this guy basically phosphorylates this guy, and this guy basically phosphorylates that guy. So they phosphorylate each other. So phosphorylation here, phosphorylation here. Okay. So, yeah, like I said, the SH2 domain is specifically like this interaction here. And so the main thing with this is just trying to show that there's many different ways the SH2 domain can be formed. That's all they're trying to get across here. And then these spots here on the receptor tyrosine kinase bind to different variations of the SH2 and SH3 domains. This gives you a bunch of examples of different RTKs, not super important. I think it's helpful to know that there are many different variations. Like, they don't all have to be the same. Like, this one, for example, is held by disulfide bonds. This one's got some cysteine-rich domains. This one's got some immunoglobulin stuff. There's a whole bunch of different variants, but it's not too important. And then here's an example of trans-autophosphorylation again. So this is something that you've already seen. So essentially the way that it works is you bind your... EGF stands for external growth factor, by the way. So yeah, RTKs are very prevalent in growth factor complexes. So usually stimulate a lot of growth in cells. So EGF here binds to this, causes a dimerization. The dimerization results in these guys becoming activated, and so this leads to phosphorylation, trans-autophosphorylation. And lo and behold, like I just said, many growth factors interact or act through RTKs. So here we have an example of this. This is going to be in the... I wish it would show me which phase it is. This is usually going to be in like G1, though, if I'm not mistaken. Because G1 is... The G phases are usually like the interphase growth sections. I'm not too sure why it doesn't specify. Oh, yeah, here. So entry into S phase is triggered. Yeah. So this is going to be in G1. So here's an example of this. So growth factors, these things will bind to two monomers of the RTK. Those two monomers will come together and phosphorylate each other, trans-autophosphorylate each other. And then that will trigger a cascade where MAP kinase is phosphorylated. And then MAP kinase is a nucleus-bound transcription factor, basically. Or it actually might not be MAP kinase. I can't remember. But MAPK then binds to parts of the DNA and triggers transcription of certain genes. And those genes result in S-phase entry, so synthesis of new DNA or replication of DNA. A bit of a sidestep from what we talked about previously. It's along the same lines, but RAS is a monomeric G protein that regulates MAP kinase signaling. And so what I want to get across with this is this idea that this process is helpful in the RTK signaling process with MAPK. So we have an inactive monomeric GTPase. You use a JEF called SOS2, which attaches a GTP, which activates the RAS. And then when you want to deactivate the RAS, you use a GAP. Cool. Some stuff you've pretty much already seen before with these JEFs and GAPs, right? So here we have a... basically what ends up happening with a lot of these RTK complexes is once you bind your ligand and dimerize and they trans-autophosphorylate, the GERB2 will bind to its specific site on the RTKs, and then that will attract the SOS, and the SOS will then act as a JEF for the RAS that is inactivated, and so it'll replace the GDP with a GTP and turn this RAS into an active form. So I think the main thing here is also to keep in mind that GERB2 binds an SH2 domain and brings SOS with it, right? So it binds to a... should I actually specify? It forms like this SH2 complex. So it binds to like this SH2 complex here, and it pulls SOS with it. So that kind of implies that these two stick together first. I'm going to be honest, I don't actually know what order they do it in. I would assume that they, based on how this is phrased, it sounds like they stick together first and then they bind together as one complex. But not too sure about that. I also was looking at this. It's kind of weird how they phrased it. Because they make it sound like... Yeah, they make it sound like the SH2 domain is on the RTK, but generally speaking, the SH2 domain is on the protein itself, I believe, not the RTK. Yeah, that's kind of weirdly phrased. So usually these proteins have SH2 domains. The the RTKs themselves don't have the SH2 domain. They have just the phosphate plus whatever amino acids that are on them. So that phrasing's a bit strange. I would double check that with the professor, how they specifically want that. But yeah, oh yeah, okay, perfect. Yeah, they do show it like this. Okay, I was concerned a little bit. Okay, anyways, so how receptor tyrosine kinases activate RAS. So here's an example of this. We bind these two dimerize, trans-autophosphorylate. The SH2 domain of an adapter protein, so a GERB2, for example, binds to one of the phosphates, and the SH3 domains of the adapter protein interact with SOS, the RAS-JEF, which the RAS-JEF then it takes the RAS, replaces the GDP with the GTP, and you get an active RAS that does a bunch of downstream stuff. So actually, this is a great image. I would copy this. So the SH2s are the ones that... So SH2s are these. Not that. SH2, SH3s. So the SH2 and SH3 specifically describe the adapter protein, so it would be like GERB2 in that case. So SH2s bind to the RTK, SH3s bind to the extra stuff. Ah, yes, here we go. Yes, okay, yeah, the MAPKs are kinases. So here we've got an example of what's going on. Here they actually show you these two are actually associated together before they bind to this. So that actually explains that pretty well. So RAS at MAPK pathway. So the MAPK pathway has cytosolic and nuclear targets for phosphorylation. So it's a couple of different cool things to note there. Basically what happens when there's no signal, everything just looks like this. There's no GTP, these are there, but nothing's active, all that jazz. When things become active, what we do is, again, bind ligand, dimerize, trans-autophosphorylate. GERB2 is able to bind to the phosphates with its SH2 domains, right? And then using its SH3 domains, it's able to bind SOS. And then SOS acts as a JEF for RAS, switching out the GTP, or GDP for a GTP. And then this RAS that is now active, because it's in its active form, is able to bind these MAP kinases. And it essentially activates these MAP triple Ks. I'm not going to say what triple K is normally. It binds MAP kinase, pushing it into its active form. When MAP kinase is active, that phosphorylates MAP kinase, which then phosphorylates MAP kinase, and then MAP kinase is the final step in this pathway, basically, where MAP kinase then phosphorylates cytosolic proteins, as well as itself goes into the nucleus to transcribe different sections of the genetic code. So this is actually a great slide to memorize or put on your note card. The insulin receptor is an RTK. Cool. That's all they're saying. So you guys know what insulin does. Insulin increases glycolysis, increases glycogen synthesis, decreases gluconeogenesis, and increases glucose uptake by cells. Basically, glucose is, or insulin is the hormone that says, hey, we have extra glucose, let's store it and use it. So, this is just an image of what the insulin receptor looks like in your textbook. It's not that important. What you should know is when insulin is not bound, it's in this inverse U conformation, and when it is bound, it's in this T conformation. That's what you should know. And usually when this binding occurs, it's actually one or two insulin molecules. It can be either one or two. It doesn't have to be just one. This is a pathway that you should know, or you should at least put on your note card. But essentially what's going to happen here is we've got pretty much the exact same stuff that we've already seen. So insulin's an RTK. Remember, these things are kind of monomeric. When insulin binds, they dimerize, they trans-autophosphorylate themselves. And then instead of having our GERB2 and SOS, we now have different things. We've got IRS1 and PI3K. These are slightly different kind of sets of machinery, but they're very similar. So again, we use our SH2 domains to bind, SH3 domain to bind to PI3K. This PI3K is a phosphatidylinositol, I can't remember the actual name, but it's like phosphatidylinositol kinase is what it is, basically. So essentially what it does is it acts to phosphorylate one of these, kind of phospholipids in the membrane. So it phosphorylates this. Initially, it's PIP2. It phosphorylates it into PIP3. Where I'm getting PIP2 and PIP3 from is I just take the PI and the P2, and I combine them. PI, P3, PIP2, PIP3. So it phosphorylates, turns PIP2 into PIP3. And then what PIP2 does, well, after being turned into PIP3, is that this thing is now a signaling molecule for PDK1, which is another kinase. And this kinase activates AKT, which is protein kinase B. And so active protein kinase B actually phosphorylates FOXO1, which is a transcription factor. And it actually inactivates it by phosphorylating it and degrades it. So here is the actual kind of showcase of what's happening here. When there's no insulin, FOXO1 is just there, and what FOXO1 does is FOXO1 actually is a transcription factor, like I said. It goes into the nucleus and it transcribes our mRNAs for PEP carboxykinase and glucose 6-phosphatase, which are both gluconeogenic enzymes. Remember, glucose 6-phosphatase, step 1, PEP carboxykinase is going back from pyruvate to phosphoenolpyruvate, remember? So these are gluconeogenic enzymes. Basically what happens with insulin is once insulin binds, we get this whole cascade, right, where PKB is activated. And when PKB is activated, it phosphorylates FOXO1. And in that conformation, ubiquitin is actually able to bind to FOXO1, which leads to degradation of FOXO1. And if FOXO1 is degraded, you can't go into the nucleus and make PepCK or glucose 6-phosphatase. So when FOXO1 is activated, glucose is made, remember, because these are gluconeogenic enzymes. But when FOXO1 is degraded, glucose gets used. It's stored. So this is actually different from glucagon and adrenaline, for example, because remember, adrenaline and glucagon, they use cyclic AMP. These do not use cyclic AMP. Insulin does not use cyclic AMP. It is specifically using these receptor tyrosine kinases, which don't use second messengers, generally speaking. Or second messengers in the form of cyclic AMP or calcium or anything like that, specifically. So now we can add to our image, what kind of goes on. So we already have glucagon or adrenaline. Remember, it goes through this pathway, does all of this. But now we have an extra thing here with insulin. So insulin dimerizes the RTKs, remember? Those RTKs then activate IRS1, and then IRS1 binds to PI3K. This thing phosphorylates PIP2 to PIP3. This then activates PDK1, which phosphorylates protein kinase B. And then protein kinase B phosphorylates FOXO1 and degrades it. And so you can see here, basically, active protein kinase B, or AKT, inhibits glycogen synthase kinase. This is a bit of a trippy one. Glycogen synthase kinase, remember, is the enzyme that phosphorylates glycogen synthase to inhibit it. So what we're doing is we're inhibiting the inhibitor. We're inhibiting glycogen synthase kinase, so this does not happen. So now glycogen synthase is actually more active. So there's two things happening here. So protein kinase B does this pathway where it somehow activates glycogen synthase. I don't think you have to know the in-depth details of how that works. I think it actually might promote transcription of more of these. I'm not too sure though. But yeah, the important thing to know is that a protein kinase B active AKT stimulates glycogen synthase and also inhibits glycogen synthase kinase. And glycogen synthase kinase normally inhibits glycogen synthase. So by inhibiting this, we're able to upregulate the activity of glycogen synthase. Which makes sense, because if you have insulin, you want to store glucose, right? So you would want glycogen synthase to be active. And another thing that insulin actually does is insulin actually stimulates glucose import into the cells. So under normal conditions, when no insulin is bound, glucose is just sitting around and nothing's going on. But when insulin binds these receptors and does the signal cascade for protein kinase B actually promotes vesicles that contain glucose transporters to fuse to the membrane. So it actually promotes fusion of these vesicles that have glucose transporters in them. And so it does this, right? It puts the vesicle into the membrane, causes these transporters to sit in the membrane, which allows for more glucose transport into the cell. And then remember, it also upregulates glycogen synthase. So it would promote glycogen synthesis or glycolysis, because remember, it inhibits gluconeogenic pathways. So it promotes glycogen synthase and also supports glycolysis, as well as the fact that it promotes fusion of these vesicles to the membrane, which allows for more glucose transport from the outside to the inside of the cell. So just a cool little thing to keep in mind. Adrenaline, glucagon promote glucose delivery, glucose to the blood. Insulin, glucose from the blood. So it's glucose to the blood for adrenaline, glucagon, and glucose from the blood for insulin. Here you have some cool little images of what happens here. So let's see, I'm trying to figure out what they are doing here. Yeah, so they're looking at glycogen synthase activity specifically. So under normal conditions, it looks like this. When adrenaline is present, right, that's the protein kinase A pathway with the phosphorylase kinase, it would inhibit glycogen synthase, remember, because adrenaline wants you to use glucose, not store it. So you'd have lower glycogen synthase activity, which is what we see. For glucagon, it's the exact same thing, right? Glucagon is also going to be the PKA GSK pathway, so it's going to inhibit it and decrease the levels. And then glycogen synthase, when in the presence of insulin, is going to go through the protein kinase B pathway, which inhibits glycogen synthase kinase, remember, inhibits the inhibitor, which leads to higher activity of glycogen synthase. So this is just a cool little thing about diabetes. Very, very prevalent in the US. So Yeah. So now we're going to talk about cyclopamine. We're going to talk about cyclopic sheep. More specifically, we're just going to talk about, like, I can't remember what the disease is called. Basically, Sonic the Hedgehog is where we're going here. Sonic Hedgehog is a protein that promotes differentiation of cells. It promotes... it's a morphogen, basically. And a morphogen, I'll talk about in a bit, is basically something, is a chemical that signals cell fate, if you will. So, in the case of cyclopic sheep, these are sheep that have one eye. And the reason they have one eye is because their parents ate this thing called cyclopamine. And so when their parents ate the thing called cyclopamine, their children had messed up development and they had one eye. And this kind of sparked the interest in the protein called Hedgehog, or SHH, or HH, or whatever the people call it these days. There's a lot of different names for it, but you can see here in normal conditions, this is what a normal embryo looks like. But when you remove Sonic Hedgehog, that's why there's 2 minuses here, you get this thing, which is not even a living being. This thing is probably dead. So yeah, very, very important. You can see here that it's pretty much used in every single limb at this point, or a major limb. So the idea is that, hedgehogs are morphogens, and their morphogens are basically just these chemicals that signal cell fate. They decide the morphology of a human being or of any being / animal / plant, whatever. You can see here, if you have too little Sonic Hedgehog, what ends up happening is you get cyclopia, which is like everything kind of converges into one line, if you will. But if you have too much Sonic Hedgehog, you actually get this kind of splitting effect, like a duplication effect. So for humans to be normally developed, they need to have just the right amount of SHH so that, you have 2 eyes, 1 nose, 1 mouth. Otherwise, if you have too little, you end up with 1 nose, 1 eye, 1 mouth, and then if you have high SSH, you end up with 2 of each, which is a bit strange. So let's talk a little bit about morphogens, so in terms of how they act and the system that they use. So here I've got for you a set of 5 cells. Great. And we're going to say that this cell right here has some sort of morphogen that it's going to release to all the other cells. So it's going to release this morphogen, it's going to release this chemical into the environment, right, or into this extracellular matrix. And eventually, that these particles or molecules are going to diffuse across, right? They're going to diffuse in different directions. In this case, let's just assume that they diffuse only this way. So they're going to diffuse to the right. And eventually you'll see a bit of a concentration gradient. So as this cell continues to produce more of this chemical, a lot of it will compile here, but entropically it's favorable to go from high to low concentration. So it'll diffuse, right? It'll continue to diffuse, but you'll have smaller amounts of the chemical here compared to here, compared to here, compared to here, compared to here, right? So the main idea is you get this concentration gradient, which is what you see here, right? So you have high levels of the morphogen over here in this area, and then as you go to the right, it gets lower and lower. And so these morphogens, what they do is they actually bind to receptors on these neighboring cells and change the fate of these cells. So these extracellular morphogens bind to these cells. Maybe they bind through an RTK, maybe they bind through some other GPCR pathway, right? But they basically bind and they result in different fates. So this dark purple cell has high concentration. It's where all of this originated. Then the cell next to it's pretty dark, the cell next to it lighter, lighter, and then this one's pretty much not affected at all. So you basically get this gradient of activity. And so because of this gradient of activity, these cells now all have completely different cell fates with different jobs. Yeah, I think that what this is trying to show you that is that, you know, so cat paws, for example, they don't really have like fully separated fingers like we do, if you will. And so that's partly due to Sonic Hedgehog mutations and stuff like that. So their paws obviously are not like our 5 fingers. They can't just like split everywhere all over the place like our fingers can. And that's because they, I believe, if I'm not mistaken, they have less Sonic. They've had less Sonic Hedgehog in that area, and so therefore the fingers kind of converge on each other. Someone should correct me on that if I'm wrong, please. And so, you know, I've talked about this. Elevated Sonic Hedgehog may cause unwanted consequences, and there's a lot of different examples of these. Autism spectrum, medulloblastomas, brain tumors, all this stuff. There's a whole bunch of things that could happen due to elevated Sonic Hedgehog. And there's also, yeah, that's pretty much it. This right here is just showing you that, funny enough, if you have elevated Sonic Hedgehog, one of the ways you can actually kind of cure it or mitigate that effect is actually using cyclopamine. Cyclopamine is a, what is it called? It is I think it's a competitive inhibitor of the receptor that Sonic Hedgehog binds to. So it actually binds to the receptor and stops Sonic Hedgehog from binding. So it's actually a pretty cool way of mitigating these. I can't remember off the top of my head if that's how that works. I believe that's what it does. But again, it's been a while since I've looked at this, so I could be incorrect on that. I'm pretty sure that's how it works, though. But here we have the signal transduction cascade, so, yeah. I would say this is one of those things that you should probably put on your note card. Let's see here. Yeah, I'd probably say put this on your note card. I'm trying to see if I want to explain this or how I want to explain this. Let's see here. Yeah, I guess I can talk about it a little bit. Essentially what's happening under normal conditions, right, is we have patched and smoothened. Hedgehog binds to this patched protein. So when there's no hedgehog, patched is actually able to inhibit this smoothened molecule, or this smoothened GPC, not GPCR, this smoothened protein. And when that's inhibited, this regular pathway happens, where this GPR 161 leads to a secondary message cascade with cyclic AMP with protein kinase A. And what that does is it takes this SUFU protein and it phosphorylates it. And when this is phosphorylated, this red thing right here is called GLI. And GLI is a repressor. Or sorry, no, wait a minute. GLI is this red one. GLI is this guy. This is, wait, no. Wait, I always mix this up. The GLI repressor is this, the red. This is the GLI repressor. And this is the this is like, it's like a repressor of a repressor. It's kind of weird. Yeah, that's how I'll describe it. So basically, it's a repressor of a repressor, if you will. So SUFU is like this carrier protein. It kind of acts as an adapter protein to carry GLI3 and this this kind of repressor thing, if you will. And so what ends up happening is when patched is active, smoothing is inhibited. And so this GPR161 is able to phosphorylate this SUFU GLI3 complex thing. And this and then what happens is proteolysis cleaves this pink part of it, and then all you're left with is this GLI repressor. And this GLI repressor, what it does is that GLI repressor goes into the nucleus and turns off a bunch of genes that are involved in splitting, basically. So it promotes being it promotes like cyclopia, if you will. And so that makes sense. So like with cyclopamine, what ends up happening is you actually end up inhibiting smoothing, if I'm not mistaken, basically. Or you actually end up binding to you bind to patch, so it always stays inhibited with cyclopamine, if I'm not mistaken. But just to reiterate what happens there, because I know it was a bit weird how I explained that. First, patched is in its active form, and when it's in its active form, it inhibits smoothened. When smoothened is inhibited, GPR161 is able to do its job normally. So it does a signal cascade that activates protein kinase A, and floating around in the cell is this SUFU GLI3 complex. GLI3 has two subunits. It's got this light pink one, or not light pink, like this like light pink one and this red one. And so what ends up happening is pKa phosphorylates the light pink subunit of this GLI3, and that phosphorylation leads to proteolysis of that pink portion. Once this thing is degraded, the GLI3 repressor, which is the red part, dissociates from the SUFU, and once that happens, this is able to go into the nucleus and repress genes that are normally there to promote separation of like limbs and stuff like that, or promote division. When Hedgehog is present, Hedgehog binds to patched, and normally patched is active without Hedgehog, and patched has the activity of inhibiting smoothened. But now, when Hedgehog is bound to patched, it's actually inactive. And because patched is inactive, it's no longer inhibiting smoothened, so smoothened is now active. So what ends up happening here is we have GLI2 instead. So GLI2 is a completely separate thing from GLI3. Once it's active, once smoothened is active, smoothened interacts with this GLI2 SUFU complex and causes SUFU to dissociate from GLI2. And then this active GLI2 basically just goes into the nucleus and turns on genes responsible for splitting of organs and limbs and stuff like that. I hope that kind of makes sense. I know I was kind of a bit convoluted in that explanation, but feel free to go back and rewind if you need a better explanation. Or alternatively, you could watch your professor's lecture. I'm sure they probably explained it way better than I did. This pathway always got me kind of confused, but I honestly think that if you just put this on your note card, you're probably going to be fine. Or at least some variation of it on your note card, you should be fine. This right here is the last slide, just a summary of transcription regulation. Already talked about this. You've got pKa, Krebs, all this stuff. Won't talk about this, but yeah. I hope this video was helpful. Again, I apologize for the last slide being super convoluted and hard to follow, but hopefully you got something out of the video, and if you did, great. If you didn't, my bad. But best of luck with studying for the exam. This is all the content I believe that's going to be on the test. You will not have Monday and Wednesday content on this exam. So again, best of luck, and have a good one.